challenges that cardiovascular disease is imposed on society. And what is important is that the atherosclerosis should not be merely seen as a disease of lifestyle and lipid or metabolic disturbances, right? So for many decades, we've primarily looked at atherosclerosis, or at least from a therapeutic side, at lifestyle changes, lowering lipid levels, introducing new types of life-saving surgery, which has all greatly contributed to reduce disease burden. But still, 47% of all death in EU is still because of cardiovascular death, right? So we should probably do better. We are already doing better. This is part of our lab. You can see how healthy and how happy we are to cycle here. We're cycling to Paris to collect money for the Dutch Heart Foundation. This is at the start where we're all still smiling. I can tell you after 4 days of sitting on a bike, we were looking a little bit different. So this is also one of the motivations to start looking into other ways to reduce cardiovascular disease, right? So maybe we should actually pay more attention to the inflammatory part of this disease, right? So it's not just lipids, it's lipids inducing inflammation and growing atherosclerotic lesions. So a few years back, we started doing more of a deep dive into these atherosclerotic lesions, and we were one of the first groups to actually use single-cell RNA sequencing to look into atherosclerotic lesions. And I hope you're familiar with this technique. It's a technique where you can basically look at the mRNA expression of every single cell in your chosen tissue. So in our case, the atherosclerotic lesion. And these were samples derived from carotid endarterectomy surgeries. So basically where you get a piece of the intima that is being removed. You can do single-cell digestion and single-cell analysis on these types of cells. And what we consistently saw, and actually many of our colleagues who did other studies, similar studies, also showed this, is that actually the majority of the leukocytes that you get out of these atherosclerotic plaques are T cells. And this is very consistent, also a little bit controversial at the time, because actually when you look at mouse models, this is much less prominent, right? So primarily macrophages are much more prominent in these lesions. But if you look at these, well, mostly advanced atherosclerotic lesions in humans, T cells dominate, and this is not just an artifact of this technique. So what we heard at the time from referees is like, well, your extraction method probably favors T cells over macrophages, and that's why we're seeing more T cells in these plaques. So we actually also use histology to actually start counting how many T cells and macrophages you find. And if you do this in human atherosclerotic lesions, you actually find that there are, in terms of numbers, there are more T cells than macrophages. But yeah, macrophages are just a lot bigger. So in terms of surface area that they cover, there's of course more surface area being covered by macrophages and T cells in plaques. So this may be one of the reasons why people always underestimated the contribution of T cells in atherosclerosis. But you can also appreciate from already this actually pretty simple picture from 2020. So in a few years' time, techniques have progressed really rapidly. Is that there's not just one type of T cell here, right? So you can see that they're already clustering into 4 different types here. So if you're going into newer techniques, and also a later publication from us and other groups also showed that actually a lot of different types of T cells in these plaques. So this makes it, of course, really interesting, right? So there's apparently heterogeneity, and there's a lot of them. So what are these T cells doing there? And so basically my lab has devoted a lot of time and effort into trying to decipher what are exactly the responses that these T cells are doing there in the plaques. And are they atherogenic, or are they perhaps protective, and which subcells are? And maybe we can, in fact, inhibit or multiply these T cells to therapeutically intervene in the disease. So one thing that already immediately struck us is when we repeated some of these experiments, but then not only included endarterectomy specimens, but also matched blood samples from these patients, is that the T cells that are circulating in the same patient look very different from the T cells that are in these plaques. And one of the most striking things was actually the expression of CD69, which we here determined with flow cytometry. So you can see that both in the CD4 and in the CD8 T cell compartment, almost half of all these T cells express CD69 compared to very few of these T cells do in the circulation. So what does that mean exactly? Well, it's CD69 is classically an activation marker of T cells, right? And there are many reasons why a T cell may upregulate CD69, but the most well-described one is directly after T cell receptor engagement on the T cell. So a T cell sees an antigen and then starts upregulating CD69. But there may be other reasons as well. So they may be exposed to some certain types of cytokines, like for instance, type 1 interferons, IL-15. Those are well-known cytokines that can upregulate CD69. But there's also one other interesting moment that T cells start upregulating CD69. That's when they become resonant memory T cells. And I actually found that really, really interesting because when I was a postdoctoral student in the US, I did not work on cardiovascular diseases at all. I worked on influenza infections and how to effectively induce memory T cells against influenza. And this was actually in the early 2010s, and resonant memory T cells were just discovered by them. So this was a really hot field back then. So how do you induce these resonant memory T cells? Because the cool thing about these resonant memory T cells is, well, as the name already suggests, they are generated and then move into a tissue and actually stay there. So they do not recirculate into the periphery. And so if you, for instance, take influenza, if you could induce a couple of these resonant memory T cells in the lungs, you would have very effective defense against a future influenza infection, right? Because you already have a few of these T cells in the exact right spots where the next infection is going to be. So I actually did quite a little bit of work on these resonant memory T cells to describe them in the lung. So basically, we immediately figured, well, what about these resonant memory T cells in atherosclerosis then? Are some of these CD69 positive T cells maybe explained by the fact that there are resonant memory T cells in the atherosclerotic lesion? And if that is the case, what are they doing there? Are they protective, or are they actually causing disease? Are these in these T cells we should be inducing or maybe reducing? So that was basically the core question that we had for this paper. It was also a major pain basically to show that these are resonant memory T cells, because resonant memory T cells are also pretty diverse. So depending on what tissue you're looking at, these resonant memory T cells look different. And this is already a relatively old review paper here. So this was basically by the end of my postdoctoral period. What we knew is that basically resonant memory T cells, they upregulate CD69, and they downregulate markers that may promote their egress from tissues. So molecules like CD62L, L-selectin, or S1P receptors, for instance, which drive them towards the bloodstream. So we, because also in papers nobody had ever described what resonant memory T cells in the vasculature look like. We had no idea what these resonant memory T cells look like, and so we also did not really have markers to use flow cytometry to see whether resonant memory T cells were there or not. So basically, we went back to our single cell RNA sequencing data sets. So the one I showed you in the previous slide, you can see there were way less dots than in this one. So this is a newer RNA sequencing data set that we generated in collaboration with the group of Gerard Pastekamp in Utrecht. And basically here you can see a plot of all the T cells that we detect in this single cell RNA sequencing plot. So basically all these T cells are derived from the atherosclerotic lesion. So if you have basically the mRNA expression of all these T cells, we hypothesized we must be able to find some kind of a cluster of T cells which bears the resemblance of resident memory T cells, at least if they are there in the plex. So we basically plotted a lot of different types of markers which are typically found in resident memory T cells from different other sites that were already described. So from the lung, from the liver, even from lymph nodes. So there's a lot of different sites where the phenotype of resident memory T cells have been described already. But the disappointing thing is if you look at all those individual genes, there's not just one single cluster of T cells popping out where you could say, well, these are probably the resident memory T cells, right? So here in panel E, we've basically added up all those signals. You can maybe say, well, it looks like a cluster 2, 0, and 1 contain well, the most hot areas for all these resident memory genes. But still, I would not find this conclusive to say, well, there are resident memory T cells in these atherosclerotic plaques. So then, in fact, actually, my PhD student, Maaike, who was the first author on this paper, she had a really good idea. She told me, well, I mean, if we already know some of these transcriptomes of resident memory T cells from other papers, why don't we look up, why don't we try to find papers where they actually did a really thorough characterization of resident memory T cells in human tissues, and try to actually insert that data into our atherosclerotic lesion data and see if we can actually find comparable T cells that would cluster together with these resident memory T cells. We found a great paper published a few years ago in Cell Reports, where they reduced a really good definition of resident memory T cells. So what they did in this paper is that they used patients who received an intestinal transplant, so a gut transplant, basically. And these people, 1 year after they received a intestinal transplant, they came back and they had a biopsy of this transplant. So basically, what happens is you can take the T cells out of the transplant and look whether those T cells were originally derived from the donor or from the recipient. So if you find T cells from the donor at that time point, like 1 year after the transplantation, well, these T cells have to be memory T cells, right? And they also have to be resident, because if they would have been circulating, we would have not been able to find them anymore after such a long time. And here you can see a flow cytometry plot of how they did this. So basically they stained for CD3, so for T cells, and they stained for the HLA type, which is not present in the patient, but only in the donor. So this is the HLA type that is specific to the donor. And then you can very nicely see that there's actually a nice population that is derived from the donor rather than from the recipient. And so they isolated these cells using flow sorting, and then performed single cell RNA sequencing. So this was basically the only paper we could find where we were really sure that people only looked at the transcriptome of a resident memory T cell in a human. And basically what we then did is we took that data and we integrated this with our data. So this is what you see. You can actually see a very small number of T cells that we got from this data set, and we inserted that into our plex. So there's two ways you can, of course, do this. You can say I'm plotting my cells, and then I'm trying to project these resident memory T cells from this other paper on them, or like we choose here, actually to completely reintegrate your data set with these few T cells from the published data set. Because when you do that, you can actually see that you change a little bit the clustering that we saw before. There's a few changes. They're the same number of clusters, but they just look a little bit different. But what is really powerful, you can see now suddenly there's a cluster 3 here, which contains almost all the resident memory T cells that we integrated from this data set. What is also really cool to see is that actually the majority of that cluster is not made up out of the T cells that we integrated. it's in fact mostly made up out of T cells from our own clustering. So this suggests that actually phenotypically or transcriptomically, these T cells are very similar to the T cells that are derived from a real resonant memory T cell population. So if we then zoom in on those T cells from our data set that clusters with the resonant memory T cells, then look at what genes they express. We can very nicely see that here that in terms of resonant memory genes, that there in fact are two genes here that are very distinct for this particular resonant memory T cell cluster. And that is ZNF683, which codes for the protein called Hobbit, and ITGA1, which encodes for CD49A, so an integrin. So knowing that, this is of course very interesting because now we actually have two genes which seems to be very good markers for resonant memory T cells, at least in humans, in the vasculature. It's also a problem because Hobbit, there are no good antibodies for it, right? So if we would just look at CD49A, probably a lot of different cells would start lighting up, right? So at this point, we actually decided to make a switch to mouse models to see if we could actually find resonant memory T cells there. And for this, we collaborated with a scientist from Lisbon, Klaas Gisbergen, who originally worked in Amsterdam, and he's actually an expert on the resonant memory T cells and has a lot of tools concerning this protein Hobbit. So he has Hobbit deficient mice, he has Hobbit inducible mice, he has Hobbit reporter mice. So we asked him, well, can you actually give us some bone marrow from these Hobbit reporter mice? So we may actually be able to detect resonant memory T cells just by Hobbit signal. And so he did. So what we did is we used LDL receptor deficient mice, which lack the LDL receptor, and therefore if you give them a Western type or high cholesterol diet, they will develop atherosclerosis really rapidly, which we actually crossed with RAG1 deficient mice. So these mice would not have any T or B cells from themselves. And then irradiated them and gave them a bone marrow transplantation with the Hobbit reporter bone marrow. So these mice nicely reconstituted their entire immune system, which the only change is that the T cells here are now going to express the TD tomato, so a red color, if they also express Hobbit at the same time. So we gave those mice Western type diet for 10 weeks, so they get nice big plaques. And then we analyzed the atherosclerotic lesion by basically digesting the aortic arch and making a single cell suspension and seeing whether T cells start to become Hobbit positive or not. And what we could see was actually a relatively small number of both CD4 and CD8 T cells that become Hobbit positive. So it's somewhere between 1 and 4% of all the CD4 and CD8 T cells become Hobbit positive in the atherosclerotic lesion. So this may be disappointing, but at the same time, it seems to also be pretty specific, because if you look at the Hobbit positive T cells, they're basically all CD69 positive, and also they shift towards a CD49 alpha positive phenotype. So indeed, it seems like CD69, CD49, and Hobbit together are also pretty good at marking resonant memory T cells in atherosclerotic mice. What is interesting is that, for instance, one of the other markers that's often mentioned as a resonant memory T cell marker, CD103, it's actually not a very good marker for resonant memory T cells. You can actually find, especially in the CD8 group, you find a lot of T cells that are actually CD103 positive, but they are not Hobbit positive, right? And we actually see this a lot when you also do flow cytometry analysis. You find a lot of CD103 positive CD8 T cells in these plaques that are also not CD69 positive, for instance, right? So I think CD103 is independently upregulated on CD8 T cells in these plaques, independent of whether they are resonant memory T cells or not. Now, the other cool thing you can do with this setup is that you actually introduce a bone marrow where Hobbit has been knocked out. So we again asked the class for some new bone marrow, but this time we did a bone marrow transplantation with mice that are Hobbit deficient and also BLIMP deficient, which is a second transcription factor that plays a major role in resident memory T cell development. So when we replaced, so this time we did a bone marrow transplantation where we replaced with either wild type bone marrow or Hobbit BLIMP double deficient bone marrow, and then again gave these mice 10 weeks of Western type diet, so we could actually monitor what happens to atherosclerosis if you basically deplete them of resident memory T cells. Now, of course, for this to work, you need to be sure that you actually depleted at least a good fraction of the resident memory T cells in these mice. So we also looked at a different site in these mice, in the liver, where normally you have resident memory T cells accumulating. And you can see that in the wild type, almost 30% of the liver T cells are CD69 positive. But when you use the Hobbit BLIMP double deficient bone marrow, you can see like about around 15% of the CD4 T cells in this case is CD69 positive, right? So we do not know whether the remaining CD69 positive T cells are resident memory T cells or antigen activated T cells, but at least we know we significantly impair resident memory T cell population by knocking out BLIMP and Hobbit in these mice. So then, of course, the major question is what happens to the atherosclerosis development. And as you may expect, actually, from knocking out such a small population of T cells in the lesion, we did not at first find major changes in how big these lesions are, right? So the plaque size of these lesions is relatively similar. However, when we were actually surprised when we looked at the phenotype of these plaques, we actually did see some subtle changes. So we saw an increase, for instance, in the number of macrophages in these plaques, so an increase in the MoMA positive area. Also, when we looked at stability markers, we saw a slight trend towards a decrease in collagen deposition, so suggesting a less stable plaque phenotype. So basically what we concluded here is that, yes, we can find resident memory T cells in both human and atherosclerotic lesions, but it's not a huge number of T cells, right? So it will not explain all those CD69 positive T cells. And we have also published other work where we actually show that there is a lot of antigen presentation in these plaques going on. So I think there's a lot of T cells actually being activated through antigen-specific means, and that's why they upregulate CD69. But it's not that they are not there. It seems like there is a small population of resident memory T cells that does reside in human and mouse atherosclerotic lesions. And I think we can at least detect them by looking at expression of Hobbit and CD49A. And the interesting thing is that our mouse studies suggest that these might not necessarily be atherogenic, as you would expect from the genes that were upregulated on these resident memory T cells. These are primarily pro-inflammatory genes. So our first idea was that probably these are atherogenic. But in fact, if we deplete them, we see the opposite result. We see actually plaque destabilization if we deplete the resident memory T cells. So, of course, I heard we should also focus a little bit on what the reviewer said and how this process went along. So we did get a lot of discussion the first time we sent this paper in, and the first one I already hoped was not going to be a discussion point, because I got a cell biologist probably as a reviewer who said, yeah, prove to me that these resident memory T cells are resident memory T cells. And well, that is just really, really difficult to do. So the classic experiment to show that a T cell is resident is do parabiosis studies, which is where you basically where you induce an immune response in one mouse, and then basically make a Siamese twin out of them with another mouse. So you stitch them together so the blood circulation is connected for several weeks, and then basically you take them apart again. And if they are resident, you do not expect them to appear in the attached mouse, but only stay in the tissue of the mouse that was originally inoculated. So I could not really see how we would handle that ethically. At least I do not know of any groups, at least in the Netherlands, who had approval for those type of experiments. And also the ideas on resident memory T cells are rapidly changing in the field as well. So people are now suggesting that actually some of these resident memory T cells do circulate. However, they also come back or basically divide, for instance, in lymph nodes and other places, and then return to tissues. So it's also not that black and white anymore, that a resident memory T cell is only maintained in that one local spot. So we try to discuss that with the reviewer, and of course they did not accept. So we had to go to another journal who would be more susceptible for our means of arguing. And there, of course, the second thing, which actually I also found a very important thing and something that we are still trying to work on, is what is the mechanism? So how do these resident memory T cells actually decrease the influx of macrophages in the plaques? And here you run into the problem that there are very few of these resident memory T cells, right? So it's really hard to study. I mean, the most logical explanation to us was that these T cells are probably, because they're pro-inflammatory, they're probably killing macrophages or killing foam cells, and in that way, basically removing some of the macrophages from the plaque. However, it could also be that they're having effects on the endothelium and maybe reducing migration of these T cells. However, so we were able to put a little bit of discussion in the discussion chapter. Basically, so for those of you who have read this paper in detail, probably have seen a little discussion on this. And this is currently ongoing work in my lab, where we're trying to see whether resident memory T cells can have direct interactions with macrophages. For instance, we're conducting spatial transcriptomic work right now, trying to confirm that hypothesis. And furthermore, there are a couple of limitations to these studies, which reviewers were also eager to point out, of course. But yeah, I think there it's really important that people always remember that models are models, right? So they do not show us everything, and every model has a limitation. So, for instance, of course, one of the limitations is that for the identification of the resident memory T cells in the plaque, we used a single cell RNA sequencing data set from the intestine. So these may phenotypically be different from those that are in the vasculature. However, what we do see across different tissues is that resident memory T cells do have a core signature that is very similar to between the different sites. So, like I said in the beginning of the presentation, a core signature of, for instance, downregulation of different migration genes and upregulation of genes that promote tissue retention, for instance. And of course, for every tissue retention is different, because there are different proteins there where the T cell can attach to, for instance. But still, there's a core gene upregulation and downregulation that we could use to actually identify these T cells. And then a couple of limitations of our mouse model. The first one is that Hobbit is not constitutively expressed in resident memory T cells, so it goes up and down. So it means that we might be underestimating the number of Hobbit-positive T cells in mice. This is true. So this is also something we described in the discussion. Actually, in the human data set, this might also be an issue because there, of course, take a snapshot of the atherosclerotic lesion, so you do not know whether all the T cells are in the same cycle as well. So at least, I think the percentages that we give is on the lower side. So there may actually be more resident memory T cells in these plaques than we detected with the Hobbit knockout model, or the Hobbit reporter model. And there are two things about the knockout model. So we know Hobbit affects NKT cell viability. So we actually also had to measure NKT cell viability in our mice, and indeed we do see a slight reduction in NKT cells, although we did not see any functional changes to the NKT cells. So they make same amount of interferon gamma, for instance, when they are Hobbit knockout. And because we know that NKT cells are probably contributing to atherosclerosis, it would also not explain why we see a reduction in atherosclerosis when we knock out the Hobbit gene. And finally, one thing where we may be able to improve on is that the Cree log system is not 100%, right? So there might be still a couple of Hobbit positive and Blimp positive resident memory T cells that have not been properly deleted from the system. So one thing we are also doing, just to be thorough, is to use complete Hobbit knockout and mice to see if we can replicate the results. So this is our team, and I highlighted the three first authors here, where I would particularly want to highlight Maaike, who just finished her PhD in my lab and is the first author on this project, and also actually came up with a lot of the ideas and of this project herself. So I think she is a very, very good PhD student. And also thanks to our collaborators, for instance, here at the HMC, which is a hospital in The Hague, where we get our patient samples from. And a shout out to Klaas van Gisbergen and co-author on this paper as well, and supplied us with a lot of important materials to look at Hobbit in resident memory T cells. So thanks for your attention, and let me know if you have any remarks or questions.
Oh, thank you so much. Amazing talk, beautiful work. We can open up if anyone would like to start with some questions for our speaker. A question? Melody, I know you have.
You can feel free to unmute yourself or write in the chat.
We have a question from Fabio. I'm not sure if the participants or the attendees can speak, or do I have to unmute?
I think they should be able to speak, so go ahead.
Go ahead, Fabio. No. Okay. Here we go.
Fabio is unmuted, but
Yeah.
He's unmuted, and I see a raised hand. Okay, then we have another raised hand here, Lydia.
Yeah, okay, yeah, good. It's okay.
Go ahead.
we just heard Fabio.
Can you hear me now?
Yes, we can.
Yes, go ahead.
Oh, okay, okay. I gave up too early. No, it's very number one. Congratulations, beautiful talk, and I'm really not into immunology, so a very naive question. So if I understand right, these cells are somehow, no, if there is, if you remove them, then you have less macrophages and less stable plaque. So these are good guys, let's say. So I understand also that not all of the markers can be easily used to trace the cells in, for instance, in humans, but can you somehow detect them, I don't know, via regular histological techniques? And if so, can you try to correlate the phenotype of the plaque? I don't know, more stable, less stable, from symptomatic, asymptomatic patients with the number of these cells?
Yeah, that's a very interesting question. So, but basically, what we've been trying to do is to see if we can then use flow cytometry to basically quantify the number of at least CD69 and CD49A positive T cells in the plaques, and then basically see if this correlates with the phenotype of the plaques, right? Whether they look stable or unstable. The problem is actually if you only do CD69 and CD49A, you do find a lot more positive T cells than I would expect from our studies here, basically, including Hobbit. So I think we need to be a little bit more scrutinous as to which markers to include to see if they are resident memory T cells or not. Indeed, we do have a cohort here of 200 atherosclerotic lesions that we've collected over the time. So we can do those studies to see whether they correlate with the phenotype. We do not have patients that are symptomatic or versus asymptomatic. So if you have an interesting cohort in your lab, then I would be very curious to know whether the resident memory T cells correlate with symptomatic and between symptomatic and asymptomatic patients.
No, unfortunately, I don't.
I'm sorry.
Sophia, you can go ahead if you unmute yourself.
Thank you so much. Thank you for a very interesting talk. It's been interesting reading the paper as well. And I was wondering how you would comment on the changes that are seen in the plasma regarding the immune cell levels after the bone marrow transplant.
Yeah, so which changes are you alluding to?
I need a second to find the figure, but I think it was in the plasma itself. It was, I think, B cells, upregulation of B cells and some other cell populations. But I remember that there were 3 or 4 significantly different immune cell populations in the plasma itself. So I'm wondering if that can influence the plaque phenotype somehow.
Yeah, so you mean so between the wild type and the Hobbit?
Exactly.
Yeah. So that could be, but we did not do a real a deep analysis of what type of B cells, for instance, changed. And of course, B cells are also not just one population. So you got the B1s, B2s, and all the different types of cells that could be protective or atherogenic. So I do not want to say that they do not play a role. The problem is, of course, if you analyze 20 or different types of immune cell populations, you're also going to find a few that are a little bit up or a little bit down. And if you just simply look at how severely those leukocyte populations are affected, I think the difference is significant. But it's not a 50% reduction in a certain leukocyte population, for instance, right? So I wonder whether those small changes really contribute to the phenotype here. But you're right, I cannot exclude it. Yeah.
Thank you so much.
Yeah.
Any other questions? I think Miron.
Yeah, thank you so much. Wow, this was like one of the top papers that we covered in the past 6 months. I mean, amount of work and complexity of the work is really amazing. So I have two questions that are not currently in any way connected. One question is if you can a little bit more emphasize on the fact that there are no really any type of links between the changes in the plasma and what you observe in plaques, because I mean, the PBMCs contain a lot of different cells, and of course, cells from the bone marrow needs to go into plaque somehow, and they're going to blood. So is it the noise of other types of subpopulations that are there and we cannot see it in the blood, or can it be something else? That's kind of first question. The second question is, after this story is being developed in the future years, and I hope it's going to be really successful, how would you target or use T cells in the therapy for atherosclerosis? I'm really kind of interested in a more like futuristic overview. What would be your approaches to really target T cells? Because as we know, in the circulation, there are many subtypes. Can we target really subspecific subtypes that are in the plaque? Should we use stents for that, or some nanoparticles, or trained T cells? I mean, I'm really, really curious what will be your comment on that. Thank you so much one more time.
Yeah, thanks. And thanks for the compliments. So your first question was basically about the changes between what we see in the plaque and in the blood, right? This actually really strikes the core of what I try to do. So because basically when there's a problem in the tissue, my first idea will be to actually also go into the tissue to see what's wrong there, right? Whereas the circulation, especially for leukocytes, is just a mixture of what is happening all over the body. Right? And there's been a lot of effort already in previous years to find certain T cell subsets, or at least markers, that are up in the circulation or down, and correlate with disease progression. So basically developing biomarkers. And it has never really worked in that respect, right? So I think what we really need to do, if we want to find subtle changes in the circulation that reflect what is actually going on in the plaque, we really need to be looking at antigen-specific T cells. So we really need to know which T cells will actually go into the plaque and do something there, right? So the number of T cells that we find in the plaque might actually also be deceiving here a bit, right? Because many of them are going to be simply attracted by the inflammation that is going on in the vessels, and actually not having a big role there. So they might be antigen non-specific. So they're going in because of the inflammation, don't see anything, and go out again for the lymphatics, right? So I think it will be up to us, and we're doing a lot of work with T cell receptor sequencing, for instance, to see which T cells are actually staying there and doing something, right? And when they leave, we might be able to detect them in the circulation. Maybe they are actually correlating with disease. So that's basically how I think that. So basically, indeed, what the circulation does, it dilutes the effect that is going on in the plaque. So you need to be very, very specific if you want to find changes in the circulation that reflect what is going on in the plaque. And your second question is actually related to that. So how do I see that we can manipulate these T cells, or actually therapeutically use them? Basically, their antigen is also going to be hugely important, I think. So there's basically two ways that we're looking at it. So you could probably work with vaccines, right? So vaccinate against atherosclerosis. So induce T cell subsets that are protective. And we may be able to use to actually deplete T cells that are pathogenic by, for instance, using CAR T cells to take them out. So those are the two main strategies that we are currently pursuing. Where I have to say, actually, the vaccination part is already a lot further than the CAR therapy, because there we need to have CARs that actually take out T cells, whereas nowadays you primarily have CAR T cells that are targeting cancer cells, right? So it's a different ball game. In terms of vaccination, we already similar strategies are being done in different autoimmune diseases, right? Basically, where they're trying to induce regulatory T cells or trying to induce T cell ignorance to certain antigens. So there's a lot of vaccination strategies already being developed, which we can more or less copy paste or at least change to the needs of atherosclerosis. Once we have defined the right antigens that the T cells are responding to in these plaques. So that's basically how we see we could potentially use T cells to manipulate this disease.
Thank you so much.
Do we have any other questions? If I may ask one thing, a little bit related to Miron's question. Again, I'm not an immunologist either. I was just wondering, as you just said, what's going on at the level of the circulation is very different from what's going on inside the actual lesion or tissue itself. Do you think that, not with respect to a vaccine, but more for prognostic methods, if these subcellular populations can be used to diagnose maybe at an early stage the initial formation, let's say, of an atherosclerotic lesion?
Yeah, absolutely. Yeah, I really believe in that. Probably not the resident memory T cells, right? Because as the name says, they're supposed to be resident, so we're not supposed to find them in the circulation. But I mean, there are clear indications that we can see changes to certain antigen-specific T cells. So also work from, for instance, Klaus Lei's lab has nicely shown that, for instance, T cells that are specific for ApoB tend to change their phenotype in the course of atherosclerosis development. So we do not completely understand how and why, but at least as a type of a biomarker, for instance, that seems to correlate. Does that mean that works for everybody? Probably not. Probably it's also not the only antigen involved, right? So we probably will need a whole panel. But at least what I think is that we can actually use those T cells indeed as kind of a rheostat. What is going on in the atherosclerotic lesion, and maybe develop it as a therapeutic tool to see who is at risk, right? So that will be the ultimate goal. Yeah.
Thank you.
I might maybe have a final question regarding the mechanisms by which do you think those resident memory cells, T cells, are contributing to stability? Do you have any speculations on how they would be protecting from atherosclerosis? Is it different types that you still need to identify that are maybe behaving differently, or what are your thoughts?
Yeah, so well, it's clear that there are different types because we see CD4 and CD8 T cell resident memory T cells. And for the CD8 resident memory T cells, because they have a cytolytic profile, right? So we see granzyme B, granzyme K, and these kinds of molecules perforin high in these cells. It would suggest some cytotoxic pathway, right? So you would think that maybe they're taking out foam cells or inflammatory macrophages. For us to know that, we would actually have to figure out what they are targeting, right? So also there we would really like to know what antigens they might be specific for. And for the CD4 T cells, it's I mean, there are papers also showing that there are cytolytic CD4 T cells in atherosclerotic lesions. So maybe they're actually killing through MHC class 2, right? So HLA class 2. Or maybe they're actually killing off some other effector T cell or B cell population. Right? So for that one is way less developed. Our ideas there, because I mean the CD8 T cells is much less complex in that respect. But we're hoping to see by actually looking at these plaques using spatial transcriptomics, for instance, to see whether these T cells are actually interacting with macrophages, or maybe even other types of cells, right? So maybe it's not even via macrophages, but through indirect means. So our hypothesis is killing right now, but they may change if we do not see any interactions between these cells.
Thank you so much. Very interesting.
If there are no further questions, thank you so much for joining us today. It was a pleasure reading your paper and hearing this talk, and a nice discussion at the end, which was very nice. Thank you very much. Thank you to all for joining. Just as a little FYI, we have our next journal club scheduled for October 14th. In the next few days, we'll come out with a link for that meeting. So please feel free to join us. Also, you, Professor Slütter, would love to have you as part of our cross action, and maybe some of your PhD students if they would also be interested. And that's it. So save the date for October 14th, and we'll come up with more info on the speaker and the paper for that. Thank you so much for presenting your work, and thank you to all for attending.
Yep.
Thanks, everyone.
Thank you.
Bye. Thank you. Have a nice week.
Hey, Christina. I'm sorry you had issues joining.
No, I was clicking the link for the October.
Oh, okay.
I had the wrong link. I don't know what.
Sorry about that. Because
No,
I mean,
it's my fault. I don't know. I October or the previous.
Yeah.
I don't know. I can't. I was on the wrong link.
Yeah. No, that's okay.
Sorry about that.
No, it's okay.
Because I could see that it was loading and it was giving me some strange message, but I said, well.
Yeah, because I was gonna email you and Fernando, because I don't know if this webinar version of Zoom is the best thing to do, because all the attendees, they cannot talk, right? I have to unmute each and every one of them automatically, and then you, even